Imagining a universe where humanity has successfully managed to scatter themselves amongst the stars. This, of course, is an idea that's very popular in science fiction and oftentimes is done through the cunning use of cryosleep. Many science fiction enthusiasts will know that this exists in many places, but of course, one of the places that it also exists that it's a little bit less well known is in the hearts and minds of researchers all over. And researchers are starting to realize that such an idea might not be quite as far as you might think, and that we might be able to put people into a state of stasis, deep sleep, or torpor in an easier way than you might think, be that through hypercooled saline solution injected intravenously, or just by careful calorie rationing and deep sleep pods. If that sounds interesting to you, then you should join us for today's episode with Aline Ingelson-Filpula, who has worked on inducing torpor in mammals and hopes to someday induce it in humans for spaceflight and other potential uses. Stay tuned for this week's episode of Sci-Fi to Reality. Aline, welcome to the show.
Thanks very much.
So let's hop straight into it. We'll toss a softball at you for starters here, but for your work now, working on human torpor, basically a version of cryosleep we can actually hopefully use someday in real life for shorter periods of time. Has science fiction inspired your work in that realm, or just in your life in general?
Yeah, I kind of grew up around science fiction, like soft, Star Wars, Star Trek. I had that phase as a kid where I wanted to be an astronaut. It began becoming a career option sort of in undergrad, where all the stuff with SpaceX building its own rockets, like that sort of started to gain steam. Thoughts of a Mars mission began becoming more like a reality instead of a long sort of future endeavor. And that got exciting for me since I'd always loved space and I would volunteer at my local observatory when I was a teenager. So I sort of knew a lot about the planets and such. I was just really excited and I thought that sort of this new frontier might be something I could get involved in. So I started taking a few Russian classes. Recent science fiction shows started to really interest me, namely The Expanse. It's my favorite. And then like I began looking into like what I could, yeah, and then I began looking into what I could actually do with it with regards to what would humans need to do to actually make sort of space travel a reality. And there's a lot of challenges to it. So really, what can't I go into research wise that hasn't been done yet? So that was kind of the start of it.
That's awesome. Yeah, Michael actually got me hooked on The Expanse, so it's funny to see that circle back around here. Um, so could you touch a little bit on how you ended up working at the observatory and what kind of work you ended up doing there to kind of get you started in your scientific path here?
Yeah, sure. So I grew up in Northern California in Chico, and the observatory was just sort of a local resource, a little bit outside of the city limits where there's less light pollution. My parents would take me there as a kid, but when I was a high schooler and community service was a thing that I knew I needed to do, I thought that would be a great, you know, resource to get into. So I was a volunteer. We had two really cool 14-inch telescopes that we could point at whatever we wanted in the sky. One had a video camera, so we could actually look at long distance objects like nebulae and other galaxies, and we would just talk to the public about them and sort of tell them what we were looking at, how far away it was, cool stories. It was really casual and laid back, but it was a lot of fun because it would often be a learning experience for me as well, and to learn from the other volunteers who had been there for years. And it just really helped me get excited about it and learn how to get the public excited about it. So it was a lot of fun.
Well, that's awesome. Yeah, I think having a casual discourse where you can start to get excited about those big grand ideas is one of the best ways to do it, and it's part of what we're trying to do here a little bit. So when you actually transitioned into college then and you started looking for how you could do research in space in general, did the human torpor path come naturally, or was that were there other steps along the way, and what got you really invested in human torpor specifically?
Well, I started out just sort of as a typical biochemistry major, and I worked in a lab actually doing sort of reproduction research, but like on a molecular level. So that was really cool to get some actual lab experience and to sort of see how a lab runs and the different techniques I could do. But in terms of like what I kind of wanted to do with my life in research, I hadn't quite pinned it down yet. But like I said, it was more sort of like the influence from the outside world that got me interested in, hey, like what if I could do this sort of thing, biology, biochemistry, but to a space perspective. So that's when I just started looking into it from that. I took some extra physics and math classes that I knew that most engineering and physics people who wanted to go into space would have to take. And I just sort of looked into what schools, sort of post undergraduate, what jobs were out there, kind of what I had to do to sort of tailor myself to actually be able to get into the space industry, so to speak.
Cool. So now that you have been working on human torpor from a kind of high level, what is the current state of the art in animals or in humans? Would you say from a relatively abstract perspective? We'll get into the biology and what's actually happening under the hood later, but from an abstract perspective, can you stick somebody in a cold tube for months on end, or an animal in cryosleep for longer? Like what is the current state of the art, so to speak?
Well, if we could do that with humans, I probably wouldn't be here right now. I'd be off doing that. But in terms of the animal side of things, which is what my lab works on, animals can actually do this already, which is why sort of what I work on in my lab is so interesting. So, with regards to freezing, there are a bunch of animals that can actually freeze themselves to last through the winter and then come back in the spring completely fine. If you try that with a human, you get a dead human. So my lab tries to look at how animals can actually do this and then apply it to humans in a slightly more non-fatal way. But the main animal that we look at, which is freeze tolerant, that's what we call it, are actually frogs. There's the wood frog, Rana sylvatica, and then there's the gray tree frog, which I just did my thesis on, Bryophytes versicolor. So they basically, in the winter, they sort of burrow under leaf litter and they flood their bodies with cryoprotectant. And in the wood frog, that's glucose, it's sugar. And then in Hyla, it's glycerol, which is kind of this sticky, gooey, it's close to sugar molecularly, but not quite. But they basically flood their bodies with this, and this enables them to build up ice around their organs, and their brain activity stops, their heartbeat stops, their blood freezes. They essentially become immobile, not even alive to any sort of biological standards. And they just stay like that for months. And then in the spring, once they thaw out, normally takes a few hours, kind of depends on the outside temperature, but then they just thaw and they're completely fine. It's really cool.
That's absolutely wild and the most fascinating experience. Oh man, I so you said it freezes around their organs specifically. So does their organs not get? Because I know that like with some cryo chamber problems, you end up getting these ice crystals that blow up blood vessels and organs, because the water in the organs expands and breaks open the organs. So does how does the frog not kill itself when freezing itself? I guess.
Yeah, that's a huge problem. And it comes at this a few different ways. One is that the frogs have specific antifreeze proteins, which, if like an ice crystal starts to form in the cell, these proteins will like panic and they'll flood to the ice crystal and bind to it and stop it from growing any larger. And then the cryoprotectant also helps. So as water starts to form outside the cells, it actually draws the water from inside the cells outwards just because of the principles of osmosis. And this actually threatens the cells with collapsing inwards and dying because you obviously can't shrink too much. So that's where the sugar comes in. It basically sort of fills the cells and it stabilizes them. It creates sort of an equal concentration environment inside the cell versus outside so that too much water doesn't flow from one side to the other. And it's essentially like a stabilizing factor. No matter the crab protectant, whether it's glucose or glycerol, or we see like other sugars and some insects that can do this, it basically just stabilizes it. So really, the crab protectants is the key for these frogs being able to do it, and it keeps ice from forming inside the cells, which, as you mentioned, would rupture the cells and cause everything to die. But yeah, if you actually, when we dissect these frogs, you can see all these crystals of ice are around the organs, but the organs themselves are still kind of soft and pliable. So yeah, it is still very true. Ice can't go inside cells; they don't play well. So that's why they sort of coordinate it to go sort of in around the organs and sort of in the body cavity itself.
So
It's
is there a
incredible.
limit to how cold they can go with that? How cold the cryoprotectants can save them?
Yeah, the frogs sort of stabilize out at around -3 degrees Celsius. Some frogs that live way up in Alaska, they can get to maybe -10. But as you get colder, like they can survive less long frozen. And then some insects can actually go all the way down to -40, but they don't actually freeze. They super cool. So they manage to keep everything liquid very, very carefully. But yeah, sort of -40 is the ultimate limit for sort of freeze avoidance, we call that. But for freeze tolerance, it's around -2, -3.
Okay.
For those super cooled insects, does that mean my understanding of supercooling is that the liquid has been cooled very carefully so that it stays liquid, but if you jostle the liquid, you will, or the liquid runs into a hard surface, it will start freezing again. Does that mean that if you take a bunch of these insects and you shake them, will they freeze and crystallize and die? And is my
Probably.
understanding
This
correct?
the insect that I mentioned. Yeah, the insect isn't an adult when it does this. It's normally the larva. So the larvae are sort of hatched in the fall. They eat everything, and then they form like a gall around themselves and their plant, and then they freeze in there for the winter. And then when they come back in the spring, they mature into adults and then mate and lay eggs over the summer, and the cycle starts again. So yeah, they're basically like in like little grubs staying completely put on a plant when they're super cooling. So
Okay,
ideally, no jostling there.
fair, fair.
Ideally.
Okay, so if we were to now transition from frogs and insects up to mammals, you also have done some work with squirrels, correct?
Yeah.
So what?
So
How do?
squirrels,
Go for it.
no, just like when you get up to mammal, well, I think I kind of knew where you were going with this. Sorry for interrupting, but once you
No,
get
no
up to
worries.
mammals, again, you can't freeze mammals. You end up with dead mammals. So what squirrels and other mammals do is they hibernate, and they hibernate in a few different ways. There's a few different types of hibernators. So obligate hibernators are ones that enter hibernation sort of regardless of the temperature, regardless of their access to food. They just sort of know when to do it. Facultative hibernators sort of enter hibernation when they're stressed in some way. So it's getting too cold, there's too little food. Um, and then when conditions are favorable again, they wake up. And then torpor is sort of more of a deep sleep state, and we see that in primates. And this is actually really interesting. I'll come back to it in a minute. But squirrels, yeah, squirrels are obligate hibernators. They eat a bunch of food in the fall, they get really fat, and then they basically sleep away the winter. And what a lot of people don't know, which I didn't know before coming to this lab, is that it's not asleep all of the time. Their body temperature firstly drops really low to about 5 degrees Celsius, but it has to wake up every few days, and during that time it shivers a lot. Its body temperature goes back up to 37 degrees Celsius, is regular body temperature. It sort of does anything it needs to do in that time, although it doesn't move around. And then it drops straight back into another, we call them torpor bout. So it basically goes through all of these spikes throughout the winter. So kind of low body temperature, really asleep at around 5 degrees Celsius. Then it shivers a lot, it spikes up to 37 for no more than a day, and then it goes back to sleep. So that's what squirrels do. And bears also hibernate, but they don't actually drop their body temperature. They keep it really warm because if you imagine how big a bear is, like the amount of energy it would take to cool that thing down would be ridiculous. So bears sort of they're warm hibernators. They stay warm throughout the entire winter. And then primates actually can enter a form of, they don't hibernate for the winter, but they can actually enter a hibernation like state in response to sort of a shortage of food. And why everyone's like, ooh, primates, is because primates are closely related to humans, right? There's sort of a larger evolutionary difference between a squirrel and a bear and a human. But when you look at primates, now we're kind of getting on the same evolutionary branch. So, of course, that's got people thinking, well, if primates can do it, would that make it easier for humans to do it? So it's really interesting to sort of start to see where this leads and the different types of hibernation in different animals and sort of start to cross-reference it.
So you mentioned both hibernation and the torpor as being slightly different. What is the exact difference there, if you don't mind explaining?
Torpor usually lasts not as long. It can last basically however long the animal wants it to, and then it can just get up and go about its business. Both of them are characterized by its metabolic rate dropping. But yeah, hibernation is more of a, what's the word for it? It's sort of like a whole process. Like it's normally like either for the winter or in response to food, and it's got like this whole orchestrated process, goes for a long time, whereas torpor is normally just like a much shorter, like sort of deep sleep state whenever it needs to happen. But they are both similar.
Okay.
And then could you expand when you mentioned the squirrel there would go about and do a couple things when it needed to every couple days? What kind of things is it doing that it needs to do every couple days? Is it just trying to get its body temperature back up so it doesn't stay in that state too long and just die and check on itself,
Yeah,
or what
it's
else is
not
going
like
on?
actually like leaving its burrow or getting up, but yeah, we assume it's basically to sort of catch up with any cell processes that, if left alone too long, it would cause the squirrel to die. So maybe it has to replicate some cells, or maybe it has to, make sure that its muscles move to keep from atrophying. Maybe it just has to make a few new really important proteins for the brain to make sure that the brain doesn't die. So yeah, it's basically just a quick warming up, sort of let the body do anything that's critical for continued survival before its body temperature drops again. Because the whole point of like the winter is that it's a really stressful time for most animals. I mean, like us humans, we have the benefits of furnaces and blankets and food whenever we want, but animals really rely. I mean, like they're sort of subject to the whims of the environment, right? So the squirrel kind of needs to conserve as much of its energy as possible to last the entire winter. So it only wakes up to do the most critical things it needs to avoid dying before sort of like going back to sleep. It doesn't have anything to waste.
Sure. So let's get into the fun stuff then. So what are we seeing with primates, and how applicable do we think some of the existing torpor that we see in primates would be to humans, I guess? Could you go a little bit more in depth in that area?
Yeah, so the primate is kind of new. We normally look at two. One is the gray mouse lemur, and I believe that one's found in Madagascar. And then the other one is called Monito del Monte. It's found in Chile. So these two are both warm hibernators. They kind of keep their body temperature high when they go into a torpor state. But really, like we just begun to look at, it's like, are the molecular mechanisms we see in the primates similar to those in the squirrels or the bears? What would be the difference between high body temperature versus low body temperature? Squirrels do the thing where they eat everything they can in the fall and be really fat, whereas torpor doesn't need that. They just kind of enter a torpor state whenever they need to. So it's really like looking at all of these conditions surrounding torpor and trying to tease out like what may be specific to the torpor itself, what may be food dependent, what may be temperature dependent. It's complicated, but you know,
Yeah,
uh, we're trying to tease it apart.
that sounds good. So okay, so you have warm hibernation and primates then. Could we go, do you know what's going on from a metabolic process and, or I guess warm bodied torpor? Well, could you go a little bit more in depth on what the metabolic process is and a warm bodied short term torpor for a primate like that might be like, for
We're still
like
kind
what the
of
cell processes are doing?
Yeah, a lot of the cell processes follow the same sort of theme throughout all the things that I've talked about, like freeze tolerance, hibernation. There's other methods of surviving extreme environments. Like they're all characterized by metabolic rate depression, which is basically the body shutting down everything it doesn't need, sort of the global downregulation of all non-essential genes and processes, and prioritizing only the critical stuff. Because again, the body assumes that it doesn't have any fuel during the winter, so it needs to survive on what it's got. So again, it's a sort of the same thing between both warm and cold hibernation. We've characterized this a lot better in squirrels so far. So I'm just going to sort of talk about what squirrels do, but a lot of this does cross over to primates. So For squirrels, actually, it's really interesting. Of course, they accumulate these huge fat stores, but they're actually insulin resistant. And a tangent that people are investigating is for type 2 diabetes, because the squirrels can just become resistant to insulin when they're super fat, and then they can be no longer resistant to insulin when they're not. And obviously, diabetes is sort of the whole your body doesn't respond to insulin anymore, and it would be great to harness this so that type 2 diabetes is no longer a thing. So insulin resistance is a main characteristic of how squirrels sort of survive with this huge amount of fat to sustain them over the winter. But when they're actually hibernating, there's a few main pathways. They switch their body from relying on carbohydrates, so like sugars, which is what humans typically start off with, to lipids, so fat, which again makes sense since they've accumulated so much fat. They prioritize proteins that prevent their muscles from sort of decaying or atrophying while they're not moving for so long, which again is a problem in humans when you look at coma patients, for example. And then also to protect their brains, they actually accumulate a lot of proteins called tau proteins. And this is what we see in Alzheimer's states, actually. Except somehow when the squirrels wake up, they're able to completely clear all these tau proteins and be fine again. So, really, in hibernation, they are able to sort of accumulate these different sort of states, which in humans are typically irreversible and lead to sort of disease states of a sort. But squirrels are able to just reverse them when they don't need them anymore. And it's a massive organizational network. Again, you can like throw a stone at basically any facet of biology and you'll find something that's different in hibernation and that needs to be teased apart. Um, it's just thousands upon thousands of things, but they all just sort of work together in order to make these different modifications and sort of reorganization of the entire metabolism to make this happen.
That's really insane that there's able to reverse. So is that gene expression reversal? Like, are we see? Is that something like, I'm going to mix up my biology keywords now, but is that I want to say methylation, but that's the wrong term. Is that change? Are they managing to do that by changing what genes are expressed in their DNA with the term that I cannot remember that is about optional biological expression? Uh, do you know what I'm trying to say?
Yeah, I do. They basically employ a lot of different things to make this happen, and I think maybe epigenetics was the term you were looking
Yes,
for.
yes, it is. Thank you.
Yeah, yeah. So epigenetics is the ability to change gene expression without altering the DNA itself, which is extremely important. And also, epigenetics is reversible, which again is extremely important for something that is not made to last a long time. So, epigenetics can fall into sort of three main categories. You've got DNA methylation, which is basically a methyl group sort of clamping onto the DNA and preventing it from being transcribed to turn into a protein. You've got histone modifications, which basically wraps up a whole section of the DNA and prevents any of that from sort of being even accessible to be transcribed. And then there is microRNA, which there is still contention over whether that counts as epigenetics. And I'm contractually obligated to say that it does count, according to my supervisor. But what microRNA do is they wait until sort of the DNA has been transcribed into messenger RNA. Then they attack the messenger RNA. They stick to it and they prevent it from becoming a protein. They either break it down entirely or they can sort of shuttle it away for later. So epigenetics is basically one whole facet of regulation that can prevent proteins from happening. Um, you can also alter the activities of the enzymes themselves. You can make them more active or less active without changing the total amount of enzyme there are. I'm sure there's others. I'm not an enzymologist, so I don't know a huge amount of detail into the whole enzymes thing. But yeah, it's sort of a coordination that can come at it from a bunch of different angles.
So, if we were to continue to go down the epigenetic route there, what? So when you see the, well, okay, in you said that there's several factors that are contributing to all these changes in the squirrels, and I just wanted to clarify here. A big part of that is due to epigenetics, then, and also, would you expect those epigenetic changes to be specific to one specific mammal, for instance? Would you expect to see the epigenetic changes you're seeing in the squirrel to also be applicable to the primate, to also potentially be applicable to a human? And if you could understand that, could you potentially do something where you could trigger the right epigenetic state, which would help clear up Alzheimer's at some distant, dreamy future, so to speak?
Yeah, 100%. And that's part of what our lab does. We compare sort of the different epigenetics, the different enzyme activity, sort of between all of these different animals and model systems which survive these different stresses. But that's all still metabolic rate depression to see where similarities and differences are. If we see the exact same thing across a bunch of animals and stresses, then we can reason that that's like super important in some way that is worth looking farther into. If there's, if it's all sort of the same in hibernation but different in freeze tolerance, we can assume that there's something specific to hibernation that it's doing, and we can look at that specifically for hibernation. And again, if everything's just all over the place, then we tear out our hair in frustration, but we say that's cool anyway, and then we can sort of look at it in a more sort of species and stress specific manner. So, like for example, DNA methylation, if we look in squirrels, we see a completely different signature than what we would see in a freeze-tolerant frog. And we even see a difference between tissues based on what are needed more. So again, I cannot stress enough the complexity of how this works. And it's not like you can just throw an epigenetic enzyme into a human, it'll do all your work for you. It would really require a lot of coordination. But yeah, we do definitely look at what might be similar between hibernation, between warm hibernation, between cold hibernation, and we hope to one day be able to sort of elucidate what's sort of key for each of these states and what molecularly is going on that allows them to happen.
That's really fantastic. It's really exciting because, yeah, hopefully one day we can start to delve further and further into the biology, yeah, biology-based world there of epigenetics and start to tease that apart. But yeah, I know it's so unfathomably complex that you need better search tools, I guess, to be able to understand what is actually causing what, I suppose.
Yeah, and again, like it's still like a new field. Like microRNAs were first discovered in like 1993. Like we didn't know they even existed before then. So we've only been able to have like 20 years to sort of look at them. For epigenetics in humans, we've been sort of looking back, and we find that they do have effects. Like for example, I read once that people in I think it was the potato famine in Ireland, their descendants have greater rates of obesity and diabetes, and they think that that's epigenetic. So again, we're sort of starting to find out like generational effects that epigenetics could have, and we now know that like epigenetics may play a role in sort of germline and reproductive cells. But again, like it's barely the tip of the iceberg. We don't know much yet. So really, yeah, it's going to have huge implications. I'm sure of it. I may be biased because I work on it, and I definitely want it to have huge implications. But so far, I think I'll still put my money on that. It's going to be another sort of important puzzle piece.
All right, let's cycle back to. I'm going to stubbornly go back to humans for a second.
Sure.
And one of the studies that I was looking at was the study of was a combination of Spaceworks and NASA's work that I shared with you here, and it looked to me, maybe I'd misinterpreted, but it looked like they had induced some form of torpor in humans for almost 2 weeks there. Um, do you have? Are you aware of any research in that field, and do you know if something like that would be sustainable? Like it seemed to me like they thought that potentially if they built the spacecraft and it could get clearance to go to Mars, they could potentially save a bunch of space and resources by inducing torpor in humans for 2 weeks at a time, and potentially you don't have to pack as much food, basically. Do you know, is that vaporware? Did I get overly excited over something like that, or do you think something like that would potentially be possible?
I mean, I read the report, and it looks optimistic and cool. What they do is therapeutic hypothermia. So basically, they flood your body with ice cold saline. And they've used this for medical interventions. The University of Maryland has used it for trauma victims. So, like, if you get shot or if you fall from a height, normally you'd have a few minutes to live. But by sort of flooding your body with ice cold saline, it sort of slows down your metabolic rate. It prevents your brain from being starved of oxygen, slows down your heartbeat, and it sort of extends your window of survival from minutes to hours for them to, do the surgery so you back up and so that you have a chance of survival. So it's therapeutic hypothermia seems to mostly have its fingers in sort of medical intervention pies right now, but they want to apply to space, which is perfectly reasonable. And what it centers around is how to sort of protect the brain. So the brain is a super finicky organ, and it's important, as we all know. If it doesn't have oxygen, you don't do too well. But what the therapeutic hypothermia does is it sort of decreases your brain's need for energy by reducing the amount of oxygen it needs and therefore the amount of energy it needs. So it can sort of it literally slows down your brain. Um, and by sort of decreasing how much oxygen, how much energy your brain needs, you can sort of stay asleep for a much longer period of time without eating or without moving around, or without doing all of your normal day-to-day activities, and that can essentially be a state of torpor. So again, yeah, the NASA report looks super optimistic, which is awesome. It would be awesome if they could pull this off, but I think a journey to Mars would take a few months. And again, if they can extend that 14-day window, that would be awesome. And currently, just my sort of concerns around it would be like, what if something goes wrong? Current therapeutic hypothermia is done under really close supervision by like doctors monitoring your vital signs and getting the concentration of saline right and keeping you at the right temperature and stuff. It's really closely regulated, whereas in space, would you have a person awake to sort of keep checking on the other people, or would it all be automated? Obviously machines break all the time, so you wouldn't want that to happen. What would the fail safe be for the hardware? Zero G, like, would it work in Super G? How would it work? So, and huge potential, and I'm excited. But those are just little things that I could think of that have to be looked into, and I'm sure people a lot smarter than me are already looking into them. So,
Right. So we've been talking about the torpor and hibernation, all of these processes slowing things down a lot, stopping non-critical processes, that sort of thing. So I want to step back. I've been curious, what are do we know what the critical processes are that need to keep going, that can't be slowed down too much or can't be stopped without killing the organism?
So things that you can sort of shut down off the bat are things like translating a lot of proteins that you kind of need to move around. For example, like moving around takes a lot of energy in the form of ATP. So if you're not moving, we switch the very energetically expensive cellular respiration pathway down to anaerobic metabolism, which requires a lot fewer enzymes. It makes a lot less energy, but you're not using it anyway, right? So you basically switch from a very expensive and complicated way of making energy into one that makes a lot less energy. It's what bacteria use, actually, but it works. Things like the cell cycle, which normally sort of turns over your cells on a day-to-day basis, that stops because that requires a lot of enzymes, and you don't really need it when you're asleep, so that stops. But certain things, it depends on the stress. So, as I mentioned in hibernation, your muscles are kind of at risk of atrophying. So, something critical might be this protein that we see called MEF2, which kind of helps stabilize the muscles, protect them from decaying. In freeze tolerant frogs, a coworker of mine found some microRNAs that help stabilize the most important neuroprotective pathways to sort of keep the brain keep what the brain needs to survive going. And then, yeah, we basically just see like a lot of like cardioprotective, so protects the heart, protects the muscles. It's basically all just these protective mechanisms to keep something from going catastrophically wrong and preventing the cells from dying. Whereas all the fluff like cell cycle and fancy energy making sort of gets put off until you need them. And digestion stops. You don't need that. Kidneys, you're not using your kidneys, so a lot of what they do stops. So yeah, it's basically just prioritizing the important stuff like brain and heart, and then shutting down the less important stuff like digestion, kidneys, what you need to move, stuff like that.
So, actually, I haven't looked into this. So, you mentioned that the muscles do actually get protected from atrophy over time in this case, or through these hibernation and such. I didn't realize that.
Yeah, and I mean, it makes sense, right? If you're asleep for, and again, this is a problem for coma patients. I mean, as far as I know, they rotate coma patients onto their sides or their backs to prevent, bed sores, and they make sure that they, flex your limbs and stuff to keep your muscles from atrophying too much. And you expect the same if like a bear is asleep for 8 months, like all of its muscles are going to degrade in a squirrel. But yeah, those animals sort of prioritize myoprotective or muscle protecting proteins to sort of keep all their muscle cells from degrading.
So
Pretty neat.
it's very cool. When you say prevent them from degrading, though, you're not working the muscle out. The muscle fibers are not breaking and then being regrown. So I would assume that this just
No,
means that your muscle fibers are not just dying and getting cleaned up. They're not just shrinking. They're just trying. They're just attempting to maintain what they have, and so you don't end up with a squirrel that can't get up
exactly.
after 3 months. Okay, so they can still get up and go get food after 3 months because
Exactly.
their
Yeah,
muscles haven't all died. Yeah, okay. So,
precisely. Which is, of course, applicable to space stuff because we see, like in current astronauts, a pretty significant loss of muscle and bone density, which is why you have to exercise for like 2 hours a day up there. So, again, there's some pretty obvious parallels to what you might need there, whether you're actually just staying awake in space or you're undergoing torpor like a squirrel or a primate or a bear might do.
it's interesting. And so just because I'm curious to learn the differences more between the squirrel and the primate and potentially us. Do you see the same when the primate goes into the state of torpor? Do you see the same behavior there with muscle retention and shorter term torpor versus hibernation, I guess?
It's a little less important for shorter term torpor, since the primate sort of isn't asleep long enough for its muscle to degrade. But we do see like sort of similar protein expression, some similar microRNAs that we know help in the sort of the muscle realm of things. So yeah, I would say that there's definite similarities.
Very cool. All right. This one is something that it's a pet question of mine. I'm very curious about anti-aging, and I read through David Sinclair's book recently, and David Sinclair has been doing his whole march around on a whole lot of podcasts talking about how to prevent aging, and he talks about how if you can put your body into an extremely high stress state, in theory, and also you put your cells down into a state where they're not consuming a lot of energy, there isn't a lot of caloric intake, then your body intentionally goes into kind of a restorative state and it attempts to work on repairing DNA and preventing degradation of cells, which means that a lot of the aging processes that we see seem to get resolved in a lot of ways. It seems to be the closest thing we've seen so far, at least according to him, to like a root cause of aging. So when I saw all of this stuff, one of my initial thoughts was, it sounds like to me, torpor or hibernation would put your body into a state that's close to that. And so I was very curious to know, would it be possible to, do you see any aging impacts on your squirrels or on primates? Specifically, I guess it would be applicable to any of the animals that are going through this. Do they live longer, or do they end up? Do they not get to find out? Might be a dark question, but might be the proper one. I don't know.
You sort of landed on the answer there. Most of the time when we do hibernation studies, we do need to sort of sacrifice them at the different hibernation time points so that we can just sort of get a snapshot of what's going on in their bodies at that time. If there are studies that have worked on like just letting an animal hibernate and seeing how long it lives in sort of different ways, I'm not aware of that yet. But I can, and it's certainly not what our lab does, but I can think of a couple interesting things. The first is a new sort of model animal that we've begun looking at, and that's the naked mole rat. Naked mole rats, wow. Naked mole rats are one of the ugliest creatures on the face of the planet, but they're one of the most interesting kind of molecularly, I think. So some cool things about them is that they can survive very little oxygen. Sometimes their burrows have, extremely high concentrations of carbon dioxide and extremely low concentrations of oxygen. So they can survive this sort of hypoxia. They don't get cancer, which is very interesting. They can live for a long time, and there's already a bunch of anti-aging work being done on these naked mole rats. They're also sort of immune to chemical pain, which again is very interesting. There's just all of these completely crazy things that naked mole rats can just do, and I'm like, whoa. So our lab has sort of begun looking at them from the hypoxia standpoint, and I think that some of the things that they're able to do just by being a naked mole rat could definitely apply to human health and space stuff. So that is kind of the wave of the future for this lab. So I could foresee some investigations into the whole naked mole rats can live a really long time and sort of have this whole anti-aging mechanism built in that we could apply to humans.
There are so many things about naked mole rats I now very much want to go research.
Right?
I
It's like I read my colleagues' like thesis that she did about it, and I was like, "Whoa, this is such a cool animal." We've like barely started working on them, and again, I don't. Yeah,
they
I
just
never
don't get
knew.
cancer.
Yeah,
They're just like, nah.
as far as I know, some other animals like don't get cancer. Like, don't take my huge one, but I think like blue whales and elephants are also pretty cancer resistant. So again, like being immune to cancer isn't unheard of in the animal world, but it certainly is interesting. I just didn't know it about naked mole rats. So
And when you say they live a very long time, do you have a number associated with that?
I would have to
It's
Google
fine
it
if
real
not. We
quick.
can we can toss it in show notes or something.
Yeah, one thing that I do know about strangely long-lived animals is actually bats. So normally the rule of thumb is the smaller you are, the faster your average metabolic rate is, but the much shorter your lifespan. So like a shrew has an extremely fast metabolic rate, but it can live maybe a year or two. Or something like an elephant has an extremely low metabolic rate, but it can live for a very long time. Bats are an anomaly. So bats are very small, sort of more around the mouse or shrew range than the elephant range, and they also fly, which also has the added it makes their metabolic rate insanely high because you need a lot of energy to fly. Yet bats can live 30 to 40 years. So they are actually really an anomaly for that general rule of thumb that a higher metabolic rate and a smaller size means you die early. So I do know that there's some anti-aging studies going on in bats to sort of see how they're able to live so long despite having a crazy high metabolic demand from being small and from being able to fly.
Sure. For what it's worth, sorry to go back for a second, but I looked it up. Mole rats live for 30 years.
Oh, dang. Okay, cool.
um, which yeah, pretty long time. And I think another anomaly according to that rule. I remember reading about that rule at some point. I don't remember what the official name is, but I remember they brought up hummingbirds because they were supposed to be another anomaly. Super small, extremely high metabolisms, extremely high heart rate. Yet they live for, I do not remember the number off the top of my head, and I'm not going to go look it up. We might throw it in show notes, but they also live for a while. Um, anyway, Michael, what's next on our list here? What should we go to next? I feel like at kind of a pretty decent spot so far.
I don't think this one is on the list, but since we're talking about long-lived animals, do any of them undergo torpor? Do you know of, or
um,
hibernation or any of that?
Bats hibernate, yeah.
That's
They
good.
form
Yeah.
these like massive roosts in caves, and then they just. I don't know a huge amount about bat hibernation. I don't think it's for the entire winter. I think it's more of a torpor-like state. Um, but yeah, bats do hibernate. As far as other small mammals, squirrels are the main one. Prairie dogs can. Arctic ground squirrels can. A lot of rodents have the capability. We've looked at Syrian hamsters who can. A lot of rodents do have the ability to hibernate. So yeah, it could be a link to your whole maybe torpor sort of slows down the aging process, and it makes sense. In humans, as far as I know, a lot of the aging research sort of centers around telomeres, which is like a long stretch of essentially junk DNA at the end of each chromosome, because all of our cells have sort of a design flaw. Whenever they replicate, a little bit at the end gets chewed off. So we have telomeres, which are basically a long scramble of junk, so that when a little bit of that gets chewed off, it doesn't really matter. And when all of that gets chewed off, and it starts chewing off important stuff, then it starts to matter, and that cell probably just dies. There's a limit to how many times cells can divide. And with regards to space, the NASA twin study, which was completed a few years back, actually found that your telomeres lengthened in space, which made everybody very interested. But when Scott Kelly came down to Earth, they shrunk back to their original length. So that's interesting, and that's probably another area of research that bears looking at. But
Interesting. Maybe you can live forever if you live in space, and you do your chores working out every day.
yeah, lots of working out, drinking bone juice, and avoiding all that cosmic radiation up there. But
Yeah, in your
yeah,
lead box.
yeah, unable to open a window. I could not. Yeah, quarantine made me have a healthy respect for the conditions that astronauts would probably have to suffer for a very long period of time. So
It's understandable. So okay, well, so I would be very curious to know what another to change topics a little bit. What's another piece of science fiction that you personally would be extremely interested to learn about? So to do a deep dive into that's totally out of your wheelhouse, so to speak.
Um, the idea of teleporters, sort of a la Star Trek style, would be really cool. Obviously, I think they'd revolutionize our way of life. Like, you could live in London, England, and work in Sydney, Australia. Commutes wouldn't be a thing. You could travel anywhere you wanted. And obviously, apparently, this has like a whole bunch of engineering and physics problems. I could barely understand anything I read about it, but it seems like we'd be breaking a few laws of quantum physics or something, and like that doesn't sound good. So that'd be cool if we got those working eventually. So
We might be able to talk to a physicist about that. That would be interesting.
Yeah, as I understand, faster than light teleportation could be that's problematic, but you might be able to do something like teleportation at the speed of light, maybe if you have a transmission line.
there seemed like some issue about the whole like deconstructing all of your atoms and like putting them back together. Like, would you be the same person? Like, how would you
Oh
like
yeah.
replicate everything exactly? Then it gets into like super physicsy things like your electron spin states, and I'm like, this is way outside of my pay grade. But it sounds like there are definitely a lot of hurdles to making that a reality.
It's outside of our pay grade too. That's why we talk to people who know more about it.
So I think we're actually at a pretty good spot here. Is there any area that you'd like? We're happy to drill down as deep as you think you could go while still managing to convey to us simpletons what's going on in your head. Is there any other piece of this, be it other aspects of your work or the you mentioned the critical care patients and the hypercooled saline solution or in general, any other ideas you'd like to drill down on more that you think you could convey to us?
Oh, it's hard. I don't know. Like, one thing that I learned the most when like sort of starting research in this area was firstly how many animals can do like insane things that we just don't know about, like between hibernation and like sort of what goes into that versus completely freezing something solid and then bringing it back to the whole. Some animals can live a long time. Bees can withstand oven-like temperatures and be completely fine with it. Like, it seems like everywhere you step, there's an animal that can do something crazy that humans only wish we could do and makes us look weak. Um, but yeah, I mean, it is hard to convey the complexity of all this. Like, it's easy to say like, oh, like microRNA might be the most important, or oh, like we should look at switching from like carbohydrate metabolism to lipid metabolism. Like, all of these definitely are things that need to be looked at, but these animals have evolved to sort of do this, and like evolution has like the greatest "if it ain't broke, don't fix it" sort of strategy ever. Like, humans have evolved to be humans, and primates, because hibernation was useful for them, like they can hibernate. And we try to look at things that aren't too different. So like, I know that there's a certain camp of scientists who think that maybe warm hibernation would be easier for a human to pull off, since if you try and lower a human's body temperature too much, you get hypothermic, and that's obviously a risk to your life. So some people think, why try to muscle a human's body temperature down to levels where it can't even go naturally? Like, let's try to keep it warm and try to apply these metabolic changes, sort of work with what we got type of thing. So again, there's just so much research being done. You can basically pick anything you want and try and figure out how different animals sort of change that to be able to hibernate, to be able to be freeze tolerant, to be able to really depress their metabolic rate and survive for long periods of time using virtually no energy. And again, it has endless applications, whether it's from like medical for surgery to space flight. Yeah, really, really, the applications are endless, and it's really cool to think about. It breaks your brain a little. And then it's difficult to convey that sense of all-encompassing applications when you're really narrowing in on one thing, which is what most scientists do. But it's really cool. I worked mostly on microRNA and a frozen frog, and like that's what I spent 2 years doing, just that.
Mhm.
So yeah, it's crazy, but I hope.
So one more question for you then. How would well, okay, two more questions, but one of them is, how would, if somebody else is really interested in what you're talking about, being able to understand all these biological processes, wants to go work on frozen frogs or human torpor or something like that, what would you recommend the path be for somebody that is super engaged and super interested and wants to be able to go chase that interest that you have?
So I didn't really know what I wanted to do in a long term. All the way up, like I knew I loved science from a young age. So like that was like the first step. I was like, okay, science. We're going to do that. We're going to make sure that we get good grades in our science classes and read. But like in undergrad, it was when I sort of had to start like, what am I interested in research wise? Like there's a lot, but I don't know what I don't know yet. So if you know what you really like, then that's great. But in terms of how I got into this, I basically just read about what I was interested in, and I looked for people who did that. And that's when I found my supervisor, Ken Story, who basically works on these animals that survive these extreme environmental stresses and figures out the molecular mechanisms of how they did it. And then I just basically emailed him saying, hey, I'm super interested. It would be amazing to like work with you for my graduate degree. And like that was it. So I'd say identifying people who work in what you're interested in and reading sort of what they do is super important, because when I was younger, I thought it was all about, like what school do you need to get into? Like what program might be best? Whereas like now it's sort of more like what people are doing the most interesting research. So again, Ken is amazing in the whole animals and molecular mechanisms thing. He's sort of a titan in the field. And then I've been sort of following researchers who work on the space side, like Afshin Beheshti is doing some amazing work on how microRNAs might orchestrate the body's response to microgravity. So I always read a lot of his papers, and I kind of see how it ties into my own work, and I just sort of keep abroad of it. So yeah, honestly, like looking, like find sort of what interests you and look at who's doing it, and then like by reading about what they do, like what their day-to-day life might be, then you can sort of identify like whether that's something you want to do. And then sometimes just like reaching out and conveying interest can make a huge difference.
Just takes a leap.
Yeah, it does, but it's paid off. So I would never discourage someone from taking a leap like that ever. So
Of course.
All right,
Well,
so I think we can move on to our last question. Is there anything you would like to promote? This could be an idea, a product, a person. Is there anything you want someone to look more into that's been listening to our show?
um, I'm not sure. Like the entire past year, I mean, as literally everyone knows, it's been interesting. It's been like the first time in, I think, pretty much living memory where like a global event has occurred, which is uniquely scientific in nature and then likewise requires science to solve. And that's manifested in both good and bad ways, which, some of the bad ways might be like how easily like misinformation can spread, like the damage it can do. A lot of the public doesn't seem to really be super aware of like what sort of public health is and what goes into that and how it's different than maybe individual health. And I get it, like it's super daunting to sort of sort through all of that. Like I can read biochemistry papers, but that's pretty much it. Like if I go into any other field, like it would be so hard to sort through. So it can seem overwhelming to even try and make heads or tails of this stuff. But like the good things that have come out of it is like you can really see like what happens when red tape is cut, and like scientists around the world can really work together to sort of arrow towards an idea and study and learn about something like a virus that has been around on this planet for less than 2 years. So really, it's been crazy. But what I'd want to promote is probably maybe just sort of more scientific literacy for everyone. So like more communication with the public on how science is already in our lives, things that we might take for granted, sort of like how the scientific method works, what peer review even is, what makes a good or a bad study, just sort of things that probably are often not addressed because science often just sort of works in the background, and then society sort of reaps the benefits in the forms of like medication or new technology, but with very little understanding of how that actually came to be. So honestly, yeah, I'd say promoting scientific literacy amongst the public and in like interesting ways that shows them how science already exists in their lives would be something that I really hope to do with my career, or as part of my career. And then I guess shows like this, where it's like you just talk about interesting things on what science can do, I think is super awesome and can help like science sort of be around in not a school environment. So that would be what I would hope to promote. That was a very long-winded answer, but
Aline, thank you so much for being on the show. We really appreciate having you.
And we look forward to hearing about your future research and any other future endeavors you embark on.
cool. Thanks so much for having me. It was a lot of fun to be able to talk with you guys about this stuff. So I hope other people find it interesting too.
Us too. And that's a wrap. So thank you so much for listening to this episode of Sci-Fi to Reality. If you haven't already, you should make sure you subscribe wherever you get your podcasts or on our YouTube channel. Our YouTube channel also has clips from a lot of the labs that we interview the researchers at. So if you want to actually see the tech that we're talking about, that's a good way to do it. We also have lots of little clips there and short form content if you're interested in that too. Finally, you can go check out show notes and links to all of our socials and everything else that you could possibly want about our show on our website, www.sci-fi-to-reality.com. That's spelled www.sci-fi-to-reality.com. So that's about it. And thank you so much for listening. Aline, thank you so much for being on the show.
We look forward
Thanks
to hearing.
for having me.
We look forward to hearing more about your research in the future.
Oh, that so failed.
Yeah, it did. This is us recording every single intro of every show.
Yeah.
Is us talking over each other 3 times before we get it right. Okay, let's give it one more shot, and if it doesn't work, it might be a
Okay.
blooper reel, or it might just cut it.